Welcome students. In our previous lecture tutorial, we have discussed transport across the cell, transport across the membrane, and we have already discussed the primary transport, how the things are being carried out from one side of the cell to the another side of the cell without any expenditure of energy and following the concentration gradient. We have already completed that part. Now in this video tutorial, we'll be discussing the active transport across the membrane. This active transport is basically of two types. Broadly, it is divided into two types. One is the primary active transport, and second is the secondary active transport. Primary active transport and secondary active transport. Let us see what is this primary active transport. Let us see what is this primary active transport. Primary active transport is a type of transport in which substances will be moving from one side of the membrane to the another side with the expenditure of energy, and the energy will be provided with the help of ATP only. This is the source of energy. And at the same time, this type of transport, we call it as uphill transport. Uphill means the things will be moving from an area of lower concentration to the area of higher concentration. Things will be moving from an area of lower concentration to the area of higher concentration with the expenditure of energy, which will come in the form of ATP. This is our primary active transport. And third thing, the primary active transport is carried out by, or it is carried with the help of some proteins which we call as carrier proteins. Carrier proteins. Carrier proteins are always involved in active transport, except permeases, which we have already discussed. Carrier proteins, it requires the participation of carrier proteins, and these carrier proteins are present on the membrane. These are the three most important things which are required in primary active transport. See, we want to shift the things. We want to make transfer of things from an area of lower concentration to the area of higher concentration. See, what is going to happen? We say this active transport, there is a I can say assumption. This active transport shows saturation kinetics. Active transport shows saturation kinetics. What does saturation kinetics mean? Saturation kinetics means if we plot a graph between on y-axis, we will take transport, rate of transport, and on the x-axis we will take the concentration absolute. What we are going to see, rate of transport versus solute concentration, we are getting a straight line, but not straight this type of line. S-shaped curve we are getting, and we call this, it shows saturation kinetics. What it means, when we increase the solute concentration, the rate of transport increases. Increase of solute concentration will increase the rate of transport. Increase in solute concentration will increase the rate of transport until a maximum is achieved. It means there is a maximum amount of, there is a particular concentration of solutes, particular amount of solutes at which the rate of transport will be maximum. If we increase the solute concentration beyond, the rate of transport is not going to increase. There is no increase in rate of transport. This we call as saturation kinetics. And this saturation kinetics tells us, the transport is being carried out by the participation of carrier proteins which are present on the membrane. How? I will just give you some example, but just a little while. See, first of all, this primary active transport requires the participation of carrier protein. For example, we have a lipid bilayer. This is a membrane, lipid bilayer. And here we have some carrier protein. This is a carrier protein present on the membrane. And again, we have lipid bilayer. This is lipid bilayer. And carrier protein is present here. This carrier protein has two binding sites. One binding site is provided for the attachment of substrate. For example, we say this is a substrate. Substrate is any substance which we want to transfer by active transport from one side of membrane to another side. Substrate is any substance which is needed to get transported across the membrane. So we have a substrate, and for this substrate there is a binding site in this carrier protein, and at the same time it has one more binding site, and that binding site is for ATP. We know in case of primary active transport, energy is provided, energy is required, which comes in the form of ATP. There are two binding sites, one for the ATP, second for the substrate. Once this ATP will bind with this, the carrier protein, for example, the same carrier protein I am taking, this ATP is binding with this carrier protein. What is the site? We call it as ATP binding site, which is present on the carrier protein. This ATP will be utilized and it will be converted into ADP and inorganic phosphate. Certainly there will be a release of energy. The energy which will be released is going to get used for bringing the substrate binding site to the exoplasmic surface. Substrate is not going to bind with the carrier protein directly. The binding of carrier protein with the substrate is a specific thing. Here, the substrate binds on the carrier protein, but it binds substrate binds on the active site of carrier protein. And this, or we can say the substrate binds on the active surface of carrier protein. It will bind on the we can say active site, or we can say it will bind at a particular site which we call a substrate binding site. This substrate binding site is not exposed. It is not exposed. It will get exposed only when there will be some energy which is required to bring the site to the exoplasmic surface. And with the result, the substrate is going to bind. And after that, there is a conformational change in substrate. For example, this is substrate. There is a conformational change in substrate. Conformational change. And what will happen? By this conformational change, the substrate will be transported to the other side of cell. The substrate will be transported to the other side of cell. This way, we can explain how active transport is being taken place. Then second part we have here, second thing we have here, which we were discussing earlier, the active transport shows saturation kinetics. We plotted the graph and we say if we want to see the rate of transport and we plot it versus the concentration of substrate, what we have seen, concentration of solute or substrate, we have seen a curve which is S-shaped, and what we predicted that as we increase the concentration of solute, the rate of transport also increases until it achieves a maxima, after which, what if we increase the solute concentration further, the rate of transport is not going to increase. And this indicates the active transport is being carried out with the help of carrier proteins. I will tell you how. For example, if we have 20 carrier proteins. This is for instance, we are just making some example. If we have 20 carrier proteins on the membrane, present on the membrane for a particular substrate. For example, if we this is the cell, we have 20 carrier proteins here, and suppose I will take two substrate molecules or two solute molecules. How these two solute molecules have to cross from this side to that side? How many substrate molecules, how many carrier proteins we have? We have 20. There is availability of 20 carrier proteins, and we have only two substrate molecules. Two carrier proteins, any two carrier proteins will come and take them instantaneously from this side to that side, right? Suppose if I further increase the concentration of substrate, initially it was 2, now it is 4. How many carrier proteins we have? How many carrier proteins, there is availability of how many carrier proteins? 20 carrier proteins. So instantaneously, we have only 4 substrate molecules, and we have 20 carrier proteins. We will take 4 carrier proteins. They will bind with this and will take it instantaneously in. The time which will be utilized for taking the 2 substrate molecules inside and the time which will be taken for carrying all the 4 substrate molecules inside will be equal. Right? And similarly, when we reach to, for example, we will take 18, 19, and even 20 substrate molecules. All the 20 substrate molecules are going to get in of the cell with the help of 20 carrier proteins, and it is not going to take any time. Instantaneously it will be doing, and we get this straight line. When we increase the concentration of substrate, we are finding the increase in rate of transport. But suppose if we have 21 substrate molecules, no 2, no 20, we have now 21 substrate molecules. How many carrier proteins do I have? I have only 20. So what is going to happen? The 20 out of this 21, we have 21 substrate molecules. 20 substrate molecules will instantaneously bind with 20 carrier proteins and will be taken in. But the last one, that is the 21st, the last substrate molecule will remain there until the carrier protein will be freed off. Once the carrier protein will be freed off, then it will come back and it will take the another one. Therefore, there is no increase in rate of transport. There is no increase in the rate of transport when we increase the concentration of substrate, and it shows saturation kinetics, which tells us something is there on the membrane which is going to transport the substances, and that something is nothing but it is carrier protein. Okay, see, the most famous example of primary active transport is sodium potassium pump. Sodium potassium pump. Again, I told you we require 2-3 things. One is the carrier protein, and here for sodium potassium pump, the carrier protein which is required is known as ATPase. It is the carrier protein. Second thing, we require energy, and that energy should come in the form of ATP. This ATP will be utilized and will be converted into ADP and inorganic phosphate. Sodium potassium pump is operational in cells, and in this pump, what is going to happen? 3 sodium ions are pumped out of cell. 3 sodium ions are pumped out of cell, and at the same time 2 potassium ions are pushed inside the cell. We can say it in this way. The carrier protein ATPase will be utilizing energy in the form of ATP, and by this energy it will be transporting 3 sodium positive ions from the inside of cell to outside, and at the same time 2 potassium ions from outside to inside. This is known as sodium potassium pump, which is operational in our cells. What is the significance of this pump? First of all, it will create a slightly electropositive potential outside. Slightly electropositive potential outside the cell. and negative potential inside the cell. For example, we have 3 sodiums out and we have 2 potassiums in. It means there is additional one sodium, additional 1 plus charge. So there will be electropositive potential outside and electronegative potential inside. Second, this pump is involved in creating the resting potential in nerve cells. In our nerve cells, resting potential is maintained with the help of sodium potassium pump. And third thing, it helps in the formation of urine. It helps in the formation of urine. One more thing, it is also involved in the maintenance of water balance. It is also involved in the maintenance of water balance between the cells and within the cell and extracellular fluid. It maintains the extracellular fluid because the sodium will move out and we have more sodium outside, more water will be in extracellular fluid, and this water is constant. Therefore it maintains the balance of water. And one more thing, in case of some marine animals, like we have seagulls and penguins, they are taking the salt water, they are consuming more sodium, and in the nasal gland, in the nasal tract, they have salt gland. They have salt gland in their nose. In this salt gland, sodium potassium pump is operational, means sodium is given out. And by the presence of this salt gland, these seagulls and penguins will excrete out the extra sodium by sodium potassium pump through this nasal gland. And the last thing, the importance of this sodium potassium pump is that it helps in secondary active transport, which we are going to discuss shortly. Secondary active transport. These are the various functions of sodium potassium pumps. There are a number of pumps which are involved in the body. We have calcium pump, we have potassium pump, we have chlorine pump. So many pumps are there. And this is what we can say about primary active transport. Let us see the last type of transport, which we call as secondary active transport. Secondary active transport. It is very simple, but we need to understand this. Secondary active transport. If we make a comparison between primary active transport and secondary active transport, primary active transport involves it requires the energy, and that energy is coming in the form of ATP. Here, it also requires energy, but it is not coming in the form of ATP. No ATP is required. However, it requires energy, but this energy does not come from the ATP. One thing. Energy is required here. This is active transport. Energy is mandatory. The energy is not going to get derived from this ATP. This is the first thing. Second thing, it will also involve the participation of proteins. These proteins will be present on the membrane, and these proteins will be present on the membrane, and they will be taking the substances, right? And involves the participation of proteins. Okay, involves the participation of proteins. And one more thing, it is a type of co-transport. Co-transport means here there is transport of two substances, two ions, two molecules at a time. Two substances are being transported. Yes, one question is there. The transport of two substances may be from outside to inside, or it may be from inside to outside, or it may be in opposite direction, depending upon the transport. Whether both the substances are moving in one direction or both the substances are moving in an opposite direction, we have two types of transport. One is symport, and second is antipode. symport and antiport. Here, if we have a carrier protein, for example, this is a carrier protein. Symport involves the transport, co-transport, involves the transport of two substances, but in the similar direction. For example, this is the direction from outside of the cell to the inside. The direction of substances is exactly same. This is symport. And antiport involves the participation of carrier protein. It involves the participation of carrier protein, which is transporting the two substances, one in this direction and second in opposite direction. Here the transport protein is involved, which is transporting two things, one in one direction and second in opposite direction. Now the question is, if the ATP is not utilized, if the ATP is not used in secondary active transport, where from energy is coming? I will tell you this way. I will try to make it very simple. I will try to make it very, very simple. See, for example, we understand there is movement of two substances. I will tell you this is a protein. This is a protein, and it will transport the substance from one side to another side. Let it be symport. It is transporting two substances. One is this, and second is this, in the same direction. For example, in case of secondary active transport, one of the substance, for example this, one of the substance will be moving from an area of higher concentration, to the area of lower concentration. A particular substance, one substance will be moving from an area of higher concentration to the area of lower concentration. Down the concentration gradient, down the electrochemical gradient. When any substance will follow the concentration gradient or electrochemical gradient, means moving from an area of higher concentration to the area of lower concentration, definitely its potential energy is going to decrease. Potential energy is going to get converted into kinetic energy. Some of the energy will be released. It is following the gradient. Some of the energy will be released. And this energy, which is released by the movement of substance from an area of higher concentration to the area of lower concentration, will be utilized for moving the another substance from an area of low concentration to the area of higher concentration. Right? Here one of the substance, it involves co-transport, involves two substances, and one substance has to follow the concentration gradient. The second substance has not to follow the concentration gradient. One substance will follow the concentration gradient, releasing some amount of energy. This energy will be utilized for moving the substance, for moving another substance from an area of low concentration to the area of higher concentration, or we can say against concentration gradient, maybe in the same direction or in the opposite direction. If it is the same direction, it is symport. If it is the opposite direction, it is antiport. Okay, one more thing. This type of substance, which is following the concentration gradient, is known as driving substance, driving ion, driving molecule, driving substance. And this type of substance, which is moving from low to high, is known as driven, because it is being driven by the driving ion. This is driving ion. This is being driven. The second is being driven because of driving one. And the two examples I will quote in case of symport and antiport. In case of symport, we will just make one illustration here. In case of symport, we have one example, that is sodium and glucose co-transport. Sodium and glucose co-transport. For example, we have a cell here. it is the membrane of the cell, and here we have some carrier protein, some protein, which is a co-transporter, and we know the sodium will be higher outside because of sodium-potassium pump. The concentration of sodium will be higher outside because of sodium-potassium pump. And the sodium is the principal ion in the extracellular fluid. Sodium is out. The concentration of sodium is more outside because of sodium-potassium pump. That's why I told you earlier, sodium-potassium pump also helps in secondary reactive transport. Sodium concentration is high. In the inside of the cell, sodium concentration is low. And the second thing is we have glucose. See, glucose is required by the cells at all times to come because it is used for energy. It is a source of energy. And this glucose is required by the cell even if it is present at low concentration in the cell, and it is present higher in the at high concentration in the extracellular fluid. But it does not matter if the glucose is very, very less here and if the glucose is very, very high here. Still, it is required by the cell. It needs to get transported to the cell, despite of the fact that its concentration is small. Its concentration is smaller in extracellular fluid. We need it. It is to be transported, whether in accordance with the concentration gradient or against the concentration gradient. We need it. Body needs it, and body has to transport it. See. For this reason, we have sodium-glucose transport. The concentration of sodium is high outside of the cell because of sodium-potassium pump. This sodium will be taken in. Down the concentration gradient by this co-transporter. The sodium will bind with the co-transporter and it will be subjected. It will be sent to the inside of the cell. It will be sent to inside of the cell following the concentration gradient, that is from higher concentration to lower concentration. Some of the energy will be released and that energy will be utilized for moving the glucose from outside of the cell to inside against concentration gradient. This is what we call as symport. Similarly, we have antiport. Let's make another example. Antiport. For antiport, we have one more example, which we call as calcium and sodium transport. It is taking place in heart cells, sodium transport. Calcium and sodium transport. Again, here we have a carrier protein. This is a co-transporter, and if we see, the concentration of sodium is higher in the outside of the cell, and the concentration of calcium outside the cell is less. What is going to happen? The sodium, and we require this calcium, the interior of the cell is requiring the calcium. What will happen? Sodium will move from an area of higher concentration to lower concentration. Following the concentration gradient, some amount of energy will be released, and that amount of energy will be used to transport the calcium, from inside of cell to the outside. Calcium will be transported out, sodium will be transported in. This type of transport is known as antiport. And in antiport transport, the sodium will be taken in. Following the concentration gradient, which will be releasing some energy that will be required to take the calcium from inside of the cell to the outside of the cell against the concentration gradient, since the concentration of calcium is very less inside and it is high outside. Still, then this transport is going to take place with the help of energy, which is released by moving the sodium down the concentration gradient. That's all about this video lecture. We'll be continuing the lectures. Thank you very much.